Hello everyone, I'm Dr. SKM Habib. In this presentation, we will be discussing about the sequence alignment technique, that is the global alignment, by using Needleman-Wunsch algorithm. Let's move on to the topic. Global alignment is part of a dynamic programming approach, which is a computational method that is applied to align two protein or nucleic acid sequences. This is done to understand the regions of similarity between the two sequences that will help us in our sequence analysis, as the dynamic programming approach is known to be providing us with the best and the optimal alignment between the sequences involved. In this approach, what we do over here is we look for the regions between the two sequences that could be kept under each other, which are identical in nature. In other words, we can say mirror images. There is also a possibility of two non-identical characters are being placed under each other, but still they having a positive score. That means these are similar characters. So in this approach of dynamic programming, every pair of characters are matched between the two sequences, and what you get, it's an alignment that consists of the matching regions, the non-matches, and also gaps. And these are the gaps which are inserted into the alignment so as to achieve as many number of matches as possible. So more the regions of match between the two sequences, more will be the clarity about the level of similarity between the two sequences. These optimal alignments provide useful information because sequence alignment forms the basis for ensuring any functional, structural, and evolutionary predictions on the basis of sequence alignment alone. Sticking to the concept of scores, now these are the ones which are utilized and applied to the alignment so as to understand and choose between the two alignments. There is a possibility in dynamic programming where you can get not only one but different types of alignments between the same two sequences. Therefore, what do we do in Needleman-Wunsch algorithm? This one came into existence somewhere in the year 1970. The method works such a way that the entire sequence is broken down into smaller fragments, and these smaller fragments are compared with each other, and what you get is the regions of similarity between the two sequences, and these broken regions or fragments are ultimately stitched together finally using the concept called Traceback technique, and that will lead us to the final optimal path. Now in this method, what we do here is we write two sequences, of which one is written across the page, the another one is written down the page in such a way that every cell occupies one character, and this character could be an amino acid character or a nucleic acid character as well. So these are also matched with each other, and there will be a matrix, as I am going to show it to you in the subsequent slides, and this matrix needs to be filled in with suitable numbers, and these are the numbers which signify whether the two characters in the intersecting cell are going to be a match or a mismatch, or if that's going to contribute to a gap. Now these gaps are added to the alignment in a manner so as to find more number of matches that could lead to more number of identical characters. More the number of gaps in an alignment, the alignment score will go down, and such an alignment is of no significance. So as you could see over here, one sequence is written across the page occupying one cell, one character. Another sequence is written down the page, and you have a matrix over here, and what you see right under the first row and right next to the first column is nothing but the gap penalty. While performing dynamic programming, we will be provided with the gap penalty. For this approach, for this specific task that we are discussing now, the scoring scheme is provided on the right hand side over here, where you can see that if the two characters in the intersecting cell are identical, then we will be providing them with 2 marks or 2 weightage. And if that happens to be a mismatch, say for example A being aligned with C, so here we can make use of -3 as the mismatch score. Likewise, if there is a gap, then we will make use of -2. Remember, gap is regarded as a penalty because these gaps are not originally part of the sequence that has been provided to us, but are inserted by the users like us just in sake of expecting more number of matching regions. So if you can see here, 0, -2, -4, -6 up to -18 on the top is nothing but the gap penalties. The difference between -2 and -4 is 2 because the gap penalty provided here is -2. And if this was -3, the gap penalty would have been 0, -3, -6, -9. So the difference between the two numbers would have been 3. So similarly, we would have followed the same scoring scheme for the other sequence that is written down the page here. So how do we fill this? Are we simply going to capture them and fill them over here, squeeze them up? No, there is a schema. Now what you see on the top left hand side corner over here is m of I comma j. Now it's nothing but this is how the algorithm works. Now what you have here is a keyword max. Here it is. We need to choose maximum of 3 values. Now what are these 3 values? Okay, so let's move on to the next slide so that we can get a better idea about it. Okay, now I can see now this is the first cell that we have to fill the matrix, and then gradually we will be moving across and down the page so as this entire matrix could be filled with appropriate numbers. So how am I going to fill 2 over here? What you have, there are 3 possibilities. Now what are the 3 possibilities? If this happens to be the cell that I want to fill, the number which is right there on top of this particular cell acts as I, because the above sequence could be treated as I, and the number that is immediately to the left of this particular cell can be treated as a J, because the sequence J is written down the page here. Okay, so if these are matches. Let us see how am I going to fill this. The formula says we need to fill this cell with one of the three possibilities, and we need to choose the value which is maximum of the three. So how is that? So I minus gap penalty, J minus gap penalty, and K plus match or a mismatch score. Now the question is, where is I? So this is my I. Where is J? Now this is my J. Then comes the question, where is K? The diagonal happens to be the K. So if I have to fill this, let us go ahead here. So let's take I. So what is the value of I? Minus 2. And what is the gap penalty? Minus 2. So minus 2 is going to get you minus 4. And this is the contribution from I. Then comes J. So here, what is the J value? Minus 2. And what is the gap penalty? Minus 2. So what is the contribution from J? Again, minus 4. Now let's go on to the third eventuality. What is the value of K? The value of K here is the immediate cell that is in the form of a diagonal is 0. This is K for the cell. So K plus match or a mismatch. So how would I know whether it is a match or a mismatch? Look at the intersecting cell and identify what character we have on top of it. We have an A in the I sequence, and we have an A in the intersecting region of the J sequence as well. So A and A are a match. It's an identical character. It's a match. What score have we given for a match? It's 2. So 0 plus 2, the answer is from K. What we are going to get over here is 2. Now as per the algorithm, we need to choose the maximum of these three eventualities, and the maximum value is 2. Where is it? Here it is. And who is the contributor? K is the contributor. So you add an arrow to point towards the contributor. Let's move on to the next cell. The next cell, in this case, if this is the cell that am I going to fill, then this character on the top becomes my I. So in this case, I is minus 4. What is the gap penalty? Gap penalty is minus 2. So it is minus 6. Then we have J. Now what do we have in J? In J, we have the immediate cell on the left is 2. And what is the gap penalty? Minus 2. So 2 minus 2 makes it 0. Let's move on with the K. So what do we have in K over here? If this is the cell that we are going to fill, the exact diagonal on top is going to be its representative K. Now the value of K here is minus 2. And what is the level of match at this intersecting region? We have a C matching with A. This is a clear mismatch. So under the circumstances, minus 2, and what is the mismatch score? Minus 3. So minus 3 minus 2 becomes minus 5. So then comes the next question. Which of these three is maximum? So which of these three is maximum? Minus 6, 0, minus 5. The answer is 0. So what do you mean by that? This 0 was contributed by J. This is how you will be filling this entire matrix by using the scoring scheme and applying the Needleman-Wunsch algorithm that is listed here on top of this slide. So you will be getting three different types of arrow marks. an arrow pointing upwards, an arrow pointing left side, and the third one an arrow that is a diagonal. If it is a diagonal, these three arrow marks helps us in the process of what we call it as traceback. Every arrow has a meaning. If it is a diagonal, we simply go ahead and make it as a matching character. If it is a arrow mark pointing towards the left, it means there is going to be an introduction of a gap at that specific position in sequence J. Likewise, in sequence I, if the arrow mark happens to be pointing upwards. So the process continues, and we make use of these arrow marks for the ultimate process called traceback. So what do we do here? This will give you a better idea. So here you can see the entire matrix is filled, and it means 8 was obtained from 6, which was obtained from 4. This is what we call it as a traceback path. And during the traceback, why do we do this traceback? We do this traceback to get the final alignment. So let me teach you traceback in a very, very crispier manner. Since it is a global alignment, which means all the characters starting from the beginning till the end are to be aligned. So we will start our traceback from the extreme right hand end bottom cell. In this case, this is the one, 8. So this 8 was obtained because of the contribution of 6, which is a clear diagonal. If it is a diagonal, so we are going to place the intersecting cells where characters under each other. So in this case, in the I sequence, what do we have? A, and in the J sequence, what do we have? A again. So the sequence on the top is I, and the sequence on the bottom is J. So A here is aligned with A. Now the traceback moves to the previous cell, which was actually the corresponding contributor to 8. Now how did we get this 6? We get this 6 from 4. And what sort of an arrow mark is it? It's a diagonal. And again, it says it's a diagonal, then we are going to place the characters at the intersecting cells under each other. So in this case, you have C, and you have C over here, and that could be seen over here. C aligned with C. Now you can see there is an arrow mark on the left hand side, and I told you in the previous slide, if it is an arrow mark pointing towards the left, it means we are going to introduce a gap at that particular position. So now you can check the third position from the last is having a gap in sequence 2, and sequence 2 eventually happens to be the sequence J, but we are retaining the character T, and this gap is what is called as, the one which is helping us to identify more number of matching regions between the two sequences. So the entire process continues until we reach 0. This approach is called as traceback technique. In order to ensure whether the matrix that we have filled is perfect or not, there is a trick in global alignment approach. The final value in this matrix, in this case 8, is 8. So this will be the overall score of the final alignment. So the final alignment score and the final value in the matrix should be identical. If it is, we can confidently conclude that our calculations are perfect, and this is a dependable score and an alignment. So a best example involving amino acid sequences. Sequence 1 is a word called similarity. Sequence 2 is a word called pillar. And in this case, we have made use of 2 for match, -1 for mismatch, and -2 for gap penalty. So the same process is continued, and ultimately what you get over here is 2 different types of alignments. Please recall, at the very beginning of this session, I had said we will get more than one alignment. And that is the beauty of dynamic programming. It will not stop with only one alignment. It tries to ensure that all the possible approaches have been utilized so that the final alignment that we get is the optimal one. There is a possibility for us to have more than one alignment. There is a possibility for us to have more than one alignment with equal score. So choosing the one which is actually biologically significant is what matters. So this is what we have learned in this discussion today about dynamic programming. These are the references that have been used. So as you can see, -2, this is the place where there is a deviation in the path of this traceback, and therefore we had 2 different alignments. Which one would we choose? We will choose the one which has the highest score. In this case, the highest score is for the alignment that is there on the top, because -1 is higher than -2. So this helps us with an understanding about the dynamic programming approach. If you need to know more about it, please follow the textbooks which are considered to be the pioneers in this field of bioinformatics: David W. Mount, Arthur Lesk, Crane and Reimer, and of course molecular modeling has nothing to do with this specific topic, but yes, it is also a great book for early career bioinformaticians. So I thank you all for your kind attention. See you again in the next video. Until then, goodbye from my side. Take care. Thank you for joining.